Hi everyone, this is exam topics covered in just 3 minutes, and in this video we are discussing the technique to detect aneuploidy. So starting from the basic technique, what we are using is the conventional karyotyping, that is using GM sustain. Then the next is like molecular methods like fluorescent probes. If we are using, then it is fluorescent in situ hybridization. So going to higher end, we can use QF-PCR, otherwise the real-time PCR. Remember, RT-PCR is not real-time PCR. RT-PCR is something which starts with the RNA. So this is neither. Then another technique that we can use is a comparative genomic hybridization, otherwise array comparative genomic hybridization or array CGH. The next technique is multiplex ligation probe amplification. So in this technique, we can detect large deletions or insertions or copy number variations, and we know that in aneuploidy there is a large deletion is there, or insertion is also possible. So these are the various techniques. Now I will show you some images where the aneuploidy detection are used. Number one, if you see this image, can you tell me what is this? Yes, this is very simple. It is a real-time PCR, otherwise QF-PCR. The next image, if I am showing, can you tell me what is this technique? That is where you can see that there is a DNA, and if you see this probe, there are two probe sequences and two primer sequences here, and these probes are ligated. So that is why this is a multiplex ligation probe amplification. It is not like a single probe that we are using. We are using multiple probes, and these are ligated later on. So that is why this is multiplex ligation probe amplification. This is something which we are using to detect large areas of deletions or insertions. So if I show you this image, can you tell me what is this? So this is array comparative genomic hybridization. Array comparative genomic hybridization means we are using two dyes. One is a red dye and the other is the green dye. Green dye is for normal and red dye is for the abnormal. When we are comparing these two genomes, so whichever areas is yellow, it means that it is equally represented in the test as well as in the patients or in the reference gene. Whereas if it is red, means there is a gain in the red region, which means that there is an amplification or duplication or trisomy. It is possible. Then if it is deletion, then what happens? For example, in the case of monosomy, in the test it will be less represented, so green will be more. So all those areas, if you get a green color, it means that it is underrepresented in the test. So this is how array comparative genomic hybridization can detect aneuploidy. So that's about aneuploidy detection techniques. Stay blessed, stay happy, and follow for more. Bye bye.